Friends, today's topic is about glutathione and its function. So what is glutathione, how it is made up inside our body, with the help of which enzyme and which amino acid, as well as which are the different important functions of this glutathione that we will look in this video. So first of all, about glutathione. So basically glutathione, that is tripeptide. Tripeptide means it is made up of the 3 amino acid, not a 3 peptide bond. So here total 2 peptide bond connecting these 3 amino acid, and that is making glutathione. And chemical name of this glutathione, that is gamma glutamyl cystinyl glycine. So this gamma glutamyl, that is indicating glutamic acid, and cystinyl, that is indicating cysteine amino acid, and lastly third amino acid, that is glycine. So this glutamic acid, cysteine and glycine together make a molecule of the glutathione. So here there is a structure of this glutathione which is also called the gamma glutamyl cystinyl glycine, means G for glutathione and G for gamma glutamyl. Why it is called gamma glutamyl? Because interesting thing is that this glutathione, which is tripeptide, but it is also important example of the pseudopeptide. Pseudopeptide means where peptide bond is not produced with the help of alpha carbon of the amino acid. Instead of the alpha, another particular molecule or carbon is utilized for the formation of this peptide bond. So here there is a gamma particular carboxyl group which is present over the gamma position of this glutamic acid is utilized, so it is considered a pseudopeptide. So simple, you can look over that structure of this glutamic acid. So here that alpha carbon, beta carbon and gamma carbon, and from this gamma carbon whatever the carboxyl group, that is utilized for the formation of the CONH, means peptide bond. So total 2 peptide bond here in this glutathione, but this gamma glutamyl cystinyl, there is a peptide bond is produced with the help of this gamma carbon of the glutamic acid. So it is called a glutathione or the pseudopeptide. Now in this cysteine there is a SH group is there. So SH, that is sulfhydryl group, that is indicating sulfur containing amino acid, but in this molecule of glutathione this SH group playing a key role for the various functions. So you have to remember this glutathione have also this SH group, and this SH group is helping in various activity. Now in detail of this, how exactly this glutathione is produced, that we are looking. So formation of glutathione. Glutathione is gamma glutamyl cystinyl glycine, means 3 amino acid, glutamic acid, cysteine and glycine. So it is generally abbreviated as a GSH. G means glutathione, SH means sulfhydryl group, which is the active group which is present in the glutathione, which is denoted or it is produced with the help of this cysteine amino acid. So it is indicating SH or sulfhydryl group. So glutamate means glutamic acid and cysteine combine and make a gamma glutamyl cysteine. So here that gamma glutamyl cysteine, 2 amino acid is combined. Further this gamma glutamyl cysteine, that will combine with the one more amino acid glycine, and that will produce finally glutathione. So glutathione, that is tripeptide, but this tripeptide contains 3 amino acid, but it has only 2 peptide bonds. So you have to remember tripeptide means not a 3 peptide bond. Now during these both the reaction, ATP is needed. So both step needs hydrolysis of each ATP. Further, you have to remember this gamma glutamyl transferase, GGT, that is important enzyme which is mostly present in the liver, which is utilized in the synthesis of this glutathione. So GGT has different 11 isoenzyme which is seen particularly in the liver. Apart from the liver, it is also present in the kidney, pancreas, intestinal cell and prostate gland. So GGT, that is also important marker for the alcoholic damage of the liver. So GGT is also utilized for the clinical practice or for the various diagnostic purpose. Now further, different important function of glutathione that we will look. So up to here we have seen that structure of this glutathione. It is produced with the help of this gamma glutamyl or glutamic acid plus cysteine plus glycine. These 3 amino acid is needed. Further, we have seen that particular SH group, that is sulfhydryl group, that is active group in this structure of the glutathione. Now which are the important function of this glutathione? So this glutathione is required for the amino acid transportation. Second, it has various co-enzymatic role. Further, RBC membrane integrity maintenance, that is another important function of glutathione. Met-hemoglobin to hemoglobin formation or keep hemoglobin in this particular state, that is also important function of glutathione. Conjugation for the detoxification process, that is also important function of glutathione. Activation of various enzyme. Apart from that, glutathione is also used by the bacteria, not by our body. In our body, folic acid is not produced, but bacteria can produce folic acid with the help of this glutathione. So glutathione is used by the bacteria for the folic acid synthesis. Further, this glutathione is also playing role in a nitric oxide formation in our body, and that can be utilized as a part of treatment in angina pectoris. So all these important function of glutathione. So one by one, each function of this glutathione and their clinical application also we are going to look in this video. So first, amino acid transport. So simple. Now how you can remember that glutathione function? So you can simply remember like ABCD. A means amino acid transportation. B, there is a B for the particular RBC membrane integrity, but C that is coenzymatic role. D, CD means conjugation and detoxification process. E means enzymatic role. So coenzyme and enzyme function, RBC and hemoglobin function, amino acid transportation and conjugation for the detoxification. So simple ABCDE of glutathione, that also you can remember. Now in this amino acid transportation, glutathione is involved in the absorption of neutral amino acid. Particularly neutral amino acid are absorbed, so it has not a particular positive or negative charge, so that all neutral amino acid that can be absorbed with the help of this glutathione across the intestine, kidney, tubules and the brain. So here you have to remember that Mr. cycle, which is already we have discussed in a amino acid transportation across the intestine. So this Mr. cycle is also working with the help of this glutathione. So glutathione is basically involved in absorption of amino acid, but here you have to remember particularly neutral amino acid. Now during this amino acid transportation, particular glutathione will be degraded and again resynthesis of glutathione will be there, and during that whole process or transportation of one amino acid across this intestine, kidney or kidney tubules or brain, there is a total 3 ATP is utilized. Now first one, that is amino acid transportation. Now second function of this glutathione, that is coenzymatic role. So metabolic role of this glutathione is mainly in reduction reaction. So basically various reduction reaction is occurring with the help of glutathione, and for that 2 glutathione molecule is needed. Now these 2 glutathione molecule that will donate hydrogen, so it will convert it into the GSSG. So GSH means indicating glutathione and SH group of the glutathione. Now such 2 molecule of glutathione that will be converted into the GSSG, and that will donate hydrogen H2. Now that H2 is utilized for the various purpose. So the hydrogen release is used for the reducing other substrate. So simple example, mallyl acetoacetate to fumaryl acetoacetate formation, which is part of the particular phenylalanine tyrosine metabolism. So during that phenylalanine tyrosine metabolism, mallyl acetoacetate is generated, and that will be converted into the fumaryl acetoacetate, and that will be further metabolized. But for this conversion, this glutathione is very helpful. For the most important molecule which is produced, that is taurine. Now this taurine is basically produced from the cysteine. So cysteine to cystic acid and cystic acid to taurine formation, and during that time this glutathione is needed. So cystic acid to taurine formation, this glutathione is playing crucial role. So taurine is basically one of the important molecule which can be utilized for the conjugation of the particularly bile acid. So already you are knowing that taurocholic acid or taurocholate, which is one of the important bile salt. So that one of the important bile salt which is produced with the help of taurine, but for the formation of taurine this glutathione is needed. So taurine is a modulator of calcium fluxes, calcium binding and movement. Apart from that, this taurine in particularly in the CNS working as an inhibitory neurotransmitter, that we will look separately in a taurine, but at present you remember for the formation of taurine this glutathione is needed. So one important function, that is amino acid transportation. Second important function, that is coenzymatic role of this glutathione. Now further important function of glutathione, that is RBC membrane integrity. So glutathione is basically used for the inactivation of free radicals. So what are the free radical which is normally produced in the cell, and that can be detoxified with the help of this glutathione. So these free radical which are generated inside the RBC, they will be generally destroyed with the help of glutathione. So this glutathione is regenerated, that is with the help of NADPH dependent glutathione reductase. Now this NADPH which is produced as a coenzyme or which is produced in the part of the HMP shunt pathway, and this NADPH is very helpful for the glutathione is regeneration. So this glutathione is regenerated by the NADPH dependent glutathione reductase enzyme. So there is a whole reaction which is converting, which is helping to make or keep this glutathione in a reduced state, that is with the help of NADPH dependent this glutathione reductase and peroxidase enzyme, that also we are going to look. At present you have to remember this NADPH which is derived from this basically G6PD shunt pathway, which is called a HMP shunt pathway or pentose phosphate pathway. So this G6PD shunt pathway, which is very helpful for the generation of NADPH, and this NADPH is basically very helpful to keep this glutathione in a reduced state. So suppose that enzyme or that particular enzyme is not present, or that pathway is not working properly, and that can lead to the one of the important clinical condition, that is hemolysis, which is typically seen in this enzyme deficiency. So hemolysis in G6PD deficiency is typically seen, and the basic reason for that hemolysis is to the reduced generation or decreased regeneration of the reduced glutathione. This glutathione, which is once generated, but it is not stay in this reduced state, and that will lead to the increased hemolysis. And apart from that, there is excessive damage by the free radical. Basically, these free radical are destroyed with the help of this glutathione, but this glutathione is not present, so free radical level is increased inside the red blood cell, and that free radical is ultimately lead to the damage of the red blood cell membrane, and that can lead to the hemolysis. So hemolysis, which is typically seen when this reduced glutathione level is decreased. So you have to remember that glutathione is very crucial molecule, or most important for destroying this free radical, otherwise hemolysis can be seen. Further, G6PD deficiency, which is that can lead to the decreased level of this reduced glutathione, which offers resistance to the plasmodium infection, which is correlate well with the malarial endemicity. So basically this malaria is occurring due to this plasmodium infection, and this plasmodium infection is less likely to seen in this G6PD deficiency patient. So why there is a connection between this G6PD deficiency and this malarial endemicity? Further, in this malaria, there is a direct connection with the glutathione. Basically, this parasite, that is plasmodium parasite, requires the reduced glutathione. Basically, this parasite needs the reduced glutathione, which is produced with the help of this G6PD or pentose phosphate pathway or HMP shunt pathway. So reduced glutathione that is not available for the survival of this parasite, so which will not be available in this deficiency of HMP shunt pathway or this particular G6PD enzyme deficiency of this HMP shunt pathway. So if that enzyme is not present, that will lead to the decreased level of this reduced glutathione. So ultimately malarial parasite is not surviving, and that will lead to the less chances for the formation of this malaria when person is suffering from this G6PD deficiency. G6 means glucose 6 phosphate dehydrogenase, which is the key enzyme of the HMP shunt pathway. Now that another important function of the glutathione. So first one, amino acid transportation, coenzyme role, RBC cell membrane integrity. Now this basically glutathione, how it is helping to reduce or destroy these free radicals. Suppose that any free radical, that is superoxide, is generated. So super SOD, that is enzyme superoxide dismutase, that will convert this superoxide into the hydrogen peroxide. Now this hydrogen peroxide will be further converted into the simple water molecule with the help of glutathione peroxidase and glutathione reductase. Now this glutathione peroxidase, that is converting this hydrogen peroxide, which is also one of the free radical, that will be converted into the water with the help of these two glutathione molecules. So simple, what happens, these two hydrogen molecules is added over this hydrogen peroxide, and that will convert this H2O2 into a water molecule. Now this glutathione that will be regenerated into the reduced state with the help of this glutathione reductase enzyme, and for the activity of this glutathione reductase, NADPH is needed. Now this NADPH that is provided with the help of these HMP shunt pathways. These HMP shunt pathway have main key enzyme, that is G6PD, that is glucose 6 phosphate dehydrogenase, and that is helping to generate NADPH. Now this NADPH is needed to keep this glutathione in a reduced state, and this reduced glutathione is very helpful to convert this H2O2 or hydrogen peroxide to convert into the water molecule, and this H2O2 is basically produced from the free radical. Suppose this molecule is not present in the reduced state, then H2O2 level will be increased inside the cell, and that will lead to the various free radical activity. So simple, you have to remember that free radical scavenging activity of the glutathione. So glutathione playing crucial role for the free radical reduction, so or reduced level of the free radical inside our body. Now basically out of this all enzyme, one particular question sometime asked, that is glutathione peroxidase, which is particularly selenium containing enzyme. So also you have learned that 21st amino acid, that is selenocysteine. So selenocysteine is particularly present in this glutathione peroxidase, that also you have to remember along with this glutathione. Now, one more important function of this glutathione to keep this hemoglobin in a particular hemoglobin state. So in hemoglobin, what happens? This Fe+2, that is ferrous form of the ion is present. But this ferrous form, if it is converted into the ferric state, then it is not available for the oxygen transportation. So Met-hemoglobin is nothing, but in Met-hemoglobin, this ion that is present in this ferric form. So Met-hemoglobin is unavailable for the oxygen transportation. So glutathione is very helpful for the reduction of this Met-hemoglobin to the normal hemoglobin. So converting this ferric state, suppose that an ion molecule which is in a ferric state, that will be reconverted into the ferrous state. So that is normal hemoglobin level will be maintained. So two Met-hemoglobin molecule that will be Met-hemoglobin that will be reconverted into the original hemoglobin or very helpful hemoglobin molecule, that is ferrous form of the hemoglobin, that is with the help of this glutathione. So it is basically very helpful to keep this hemoglobin in actual state or convert this Met-hemoglobin to hemoglobin. So that is also important function of the glutathione. Further, another function that is of glutathione, that is conjugation for the detoxification. So various endogenous and exogenous substances are that, and that need to be excreted out, and that is possible with the help of conjugation process. So various conjugation process, that is called as the second phase of the detoxification. So one of the important molecule which is utilized for the detoxification, that is glutathione. So glutathione helps to detoxify several compounds by transferring this cysteine group. So that is GST. So that is another short form of for the conjugation. For that this glutathione is utilized. So glutathione S transferase, that is short form of the GST, glutathione S transferase, and that is helping to detoxify these various exogenous substance like OP poisoning, means organophosphorus compound or halogenated compound. Apart from that, nitrogenous substance and heavy metal that also destroyed or detoxified with the help of this conjugation process with the help of glutathione. So here simple that cysteine group is transferred, and that is transferred with the help of that GST enzyme. This GST have also different 6 isoenzyme that also we will look separately, but at present you have to remember this glutathione is basically utilized for the conjugation process for the detoxification purpose. So various drugs are also metabolized with the help of this conjugation process with the glutathione. So that various function, amino acid transportation, then second coenzymatic role, then also that Met-hemoglobin to hemoglobin conversion and conjugation for the detoxification. Now one more function of this glutathione, that is activation of enzyme. So many enzymes having SH group in the active site. So basically this active site or site where particular substrate is attached over the enzyme, that is called active site. So papain, urease, succinate dehydrogenase, glyceraldehyde 3-phosphate dehydrogenase, these are the enzyme where active site have the SH group, and this SH group are kept in the active form with the help of this glutathione. So basically this glutathione is very helpful to keep this enzyme in an active form or to keep their active site in a present, or otherwise what happen, this active site will be masked and that will lead to the reduced activity of the enzyme. So this enzymatic activity will be remain present with the help of this glutathione. So such enzyme are remain active in the reduced form only, otherwise that enzyme will not be in an active form. So that keep enzyme in an active form, that is also another important function of the glutathione. So apart from that, glutathione which is present or which have various role in our body, this glutathione is also used by the bacteria for the folic acid synthesis. So simple, what is the name of this folic acid? That is steroyl glutamic acid. Now this glutamic acid is mostly derived with the help of this glutathione. So glutathione which is taken by the bacteria, and that bacteria is basically utilize this glutathione for the folic acid formation, and folic acid is very important molecule for the bacteria for the cell wall formation. Now most important clinical application of this glutathione, that is this particular nitric oxide formation. So this N-acetylcysteine which is considered as a glutathione precursor. So N-acetylcysteine which is also one of the precursor molecule for the glutathione formation. So it protects nitric oxide from being metabolized by free radical scavenger as well, and enhances the nitric oxide activity. Basically what happens, nitric oxide that is produced with the help of nitric oxide synthase enzyme, but this nitric oxide synthase formation, that enzyme that will work over the particular substrate, but suppose that precursor molecule, that is glutathione precursor, that is N-acetylcysteine, over that this particular nitric oxide is protected. So it protects nitric oxide from being metabolized by the free radical scavenger, as well enhances the nitric oxide's activity. Basically this N-nitroglutathione is a nitric oxide donor. So basically nitric oxide is donated with the help of S-nitroglutathione molecule. which inhibit the platelet agglutination. Platelet agglutination means simple platelet aggregation will be inhibited with the help of this S-nitroglutathione molecule. So basically this S-nitroglutathione, which is a nitric oxide donor and which is very helpful for the inhibiting the platelet agglutination. And further, one more thing you have to remember, N-acetylcysteine, that is also working as a precursor of the glutathione. So is a glutathione precursor. Now what is the clinical application? Basically angina pectoris. So where drug is given, that is nitroglycerin. So this glyceryl trinitrate or the nitroglyceride, which is requires the glutathione for the production of nitric oxide. So ultimate goal of this treatment, that is production of nitric oxide, where the patient is suffering from the angina pectoris. So if nitric oxide is produced, that will dilate the coronary artery, so and that will improve the blood circulation surrounding the heart. So coronary artery dilation, that will very helpful to relieve this angina pectoris. So basically the main purpose of this and providing of nitroglycerin, that is production of nitric oxide, but this nitric oxide is basically produced with the help of this glutathione. So one of the important clinical application or clinical use of this glutathione, that is production of nitric oxide with the help of this nitroglycerin, and that can be very helpful particularly in emergency situation of angina pectoris, and that dilating of coronary artery, that can be very beneficial for the patient suffering from the angina pectoris. So there are various functions of glutathione. So you have to remember that simple A, B, C, D, E. A for the amino acid transportation, B for the RBC membrane integrity, C means coenzymatic role, conjugation for the detoxification process, E means enzymatic activity. So these all RBC as well as hemoglobin. So whenever you are writing RBC, you have to remember about Met-hemoglobin to hemoglobin formation. So these are the different functions of this glutathione and their important role in our body.